stroke animal model. We have actually conducted phase 2 clinical trial spinal cord injury using acetic FGF, Fibrin cocktail on very, very chronic patients. And we have actually concluded the result. Actually, the result is positive. Comparing the historical review with Fishbone, almost 1,000 patients fully followed. And we really see the benefits using that kind of treatment application. However, actually we report to the health department and we are waiting for the conclusion from the committee. So I'm not allowed to contact with any kind of place to release the result. So it is quite soon to be concluded in the health department, and hopefully within 1 month I'll be presenting data in the place. Thank you. So today I will change my talk to stroke using the stem cell. So this is another work in my lab. So as you see, this is also a major problem in neurological care. Actually, the population of stroke is much bigger than in spinal cord injury, and 85% is due to ischemia, due to majorly the cerebral vascular occlusion, cerebral vascular disease, including the cerebral ischemia, called Infarct, and TIA. So if you look at the lesion side of stroke, there is a core, which is the area with dense ischemia, and a neighboring moderate ischemia, which is called penumbra. And this is our previous data with neuroprotection in acute stroke model in animal using resveratrol neuroprotective agent, which you can see 1 week after that, if there is no treatment, there is atrophy of the involved cerebral cortex, and you treat it, it is some neuroprotective effect. And the process of the strategy of stem cell transplantation includes the extraction from the bone marrow, and then isolation, and then do the injection to the infarcted area. We have doing many kind of research and find a way to do it which can be safely applicable in a clinical situation. That means we don't use bone fetal capsaicin or any animal feeder. So which is more safe to prevent any so-called some Hualien disease or animal related disease when in concern of clinical application. So we have actually applied to US patent on this. So this is our new know-how to use serum-free, animal serum-free, and animal feeder-free in the process of human bone marrow stem cell. So this is a major process. And the adult stem cells we can harvest from the bone marrow, which is we'll do the autologous bone marrow transplantation, which carries the benefit of preclude any kind of immuno rejection and ethical problem. This is our data to use a special system to homogenize the stem cell, which is called size-sieved, which is with a kind of special sieve, and then pouring all this bone marrow stem cell into it, and further to proliferate and to produce a homogeneous clone before using that as a transplantation. And that has been published in 2002 by my colleague Hong Shijie. And there are also, during the induction that time, we use beta-methyl and retinoic acid and other growth factor to differentiate the bone marrow stem cell into neurons. And later, in a later paper, we use other molecule which is safer in clinical use to differentiate into neuron, because this too is carcinogen, and this is neurotoxic, and this carcinogen, which may carry some risk during the clinical trial. However, in that time, since that's an animal experiment, we use that to differentiate the bone marrow stem cell into neuron-like cells, which you can see the bipolar process after induction. And we test this cell with Western blot, which carries neuroprotein, for example, nestin, NSE, and UN in the early induction. Later, when the neuron-like cell become more mature, it expresses neurofilament, but not the protein expressing the early neuronal status. With the patch frame, we test this kind of cell that carries the calcium channel, and sodium channel, potassium channel, which is depolarization. The membrane carries the property of neuron-like property. So we conclude that it's possible to differentiate in vitrally. The bone marrow stem cell can differentiate into neuron-like cells. And we transplant this cell into the brain and also spinal injury model, which is a NYU impactor. The inventor is here. And however, as I said, the beta-methyl pretreatment is toxic. In the transect spinal cord, when we transplant this kind of cell, some red dies, and in the stroke model, the middle cerebral artery occlusion model, 50% die in a preliminary experiment. So we try to find some other molecule which is safer to differentiate the human bone marrow stem cell into neuron-like cells. There are three. We test a bunch of molecules, but we conclude three which is more safer to use, including GDNF, PCAP, pituitary adenosine activating polypeptide, and deuterial cyclic AMP. And this is a paper we have published, and you can see with the GDNF PCAP, the cell can be differentiated into neuron-like cells, expressing neuromolecular like a synapsin. You can see the process of the neuron-like cells expressing synapsin 1. And also, it expresses a neural intermediate filament alpha intermixing with GDNF differentiate or PCAP differentiate. So this is the inducer we are now using to differentiate the bone marrow stem cell into neural cells before the transplantation in an animal experiment. The GDNF or PCAP treat bone marrow stem cells carries the characteristics including they have elongated cell process and increased process branching. They increase neurofilament L protein. They increase cytoskeleton protein alpha tubulin. They increase vascular protein synapsin 1. They increase neurogenital marker alpha intermixing, which is quite promising. But transplantation of bone marrow stem cell want to prolong the local effect. We have using several methods to have the prolonged local effect. First, the delivery method, direct injury to injection into the infarction site, but prevent the gush out and keep them in the injection area by the application of fibrin glue on the surface of the cortex. Also use other growth factor together with fibrin glue, which is a strategy used in the spinal cord repair, which has been published in 1995 in Science. And we test several behavior methods. So this is the animal we use, which is long even rats. And this is a condition of surgery, which is was the craniotomy and produce MCA ligation, and then with ligation of right middle cerebral artery, as you can see here. As you can see, this is before the ligation. It's a middle cerebral artery down to the very proximal end, and then ligated with 10-0 suture. And we, after MCA occlusion, we measure different parameters, including the infarction burden, and we do neurobehavior tests, including rotary test, cross-beam power test, and windshield region test. This is the situation. After dissection, you can see the whole brain, and we do it particle slice, and then stain with TTT stain to measure the infarction area. And this is the brain matrix slicer. And using the AIS software to measure the infarction area, and using the formula to calculate the total infarction burden by this formula. And this is the treatment protocol. Because we found most of the stroke patient, if that is treatable, that can be rescued by the in the first 8 hours or first day by injection of thrombolytic agent. If the patient cannot be rescued, they send out to the neurology ward and waiting for the rehabilitation. So the major patient, the target population is the patient cannot be rescued by the tissue thrombotic protein, and that patient waiting for the new therapy. So for the design of animal experiment, we should focus on this patient. We should not do the animal just after the stroke and do the therapy. We should do the stroke and wait to the subacute and chronic stage and rescue it. That is more similar to the clinical situation. So the protocol is like this. We do ischemia insult with MCA ligation and CCA clipping, and do the reperfusion entry, and waiting, and then 1 week later we do the rescue. With different kind of treatment, including bone marrow stem cell only, bone marrow stem cell fibrin group. This is the first stage, and later we will add growth factor together with bone marrow stem cell, and then 1 week after the MCA ligation and reperfusion injury, and later we do measurement, including the infarction, forelimb, and motor behavior testing for 4 weeks. So this is the result after the focal cerebral infarction, ischemic injury. As you can see, the control, this is a sham. The control without treatment, you can see the atrophic change of the involved cerebral cortex. If you treat it with bone marrow stem cell, you can see the better morphology than the control. Adding the fibrin group to prevent the leakage of the bone marrow stem cell from the transplantation site seems to have some more beneficial effect than the bone marrow stem cell only. And this is a lateral view of the animal brain. This is a bone marrow stem cell only, and this is a bone marrow stem cell together with the fibrin group. And this is in both sides. You can see the control bone marrow stem cell only, and bone marrow stem cell together with fibrin group treatment. The infarction size significantly reduced when treating with bone marrow stem cell together with fibrin group. The infarction volume is reduced to 60% of the untreated or controls. The behavior test, the first one is a roll test. As you can see, the animals receiving the bone marrow stem cell treatment group doing the roll test is a longer latency compared to other group, including bone marrow stem cell only, control and shape group. Grasping power test is the method to let the animal to grasp the handle and test the grasping power for 10 times and pick up best 3 times, and to have an average of 1 trial, and then calculate that and compare that statistically. This is an effective forelimbs, which is no difference between groups. And this is the grasping power test of the effective forelimbs. As you can see, the animal received bone marrow stem cell fibrin group transplantation. It's significantly improved the grasping power at 2 weeks, 3 weeks, and 4 weeks after the transplantation. And we'll test the transplant cells, which stand with the human nuclear DNA in the bone marrow stem cell group group, you can see the cells. Put in human, because we transfer human cell into the rat brain, so this has been proved that in the tissue we have the DNA, human DNA tissue. In the bone marrow stem cell, because of probably the leakage, you see the DNA is almost nothing. So this is an easy method, and using Fibrin group containing bone marrow stem cell to rescue the infarction of the brain, which is quite easy to apply if that is a clinical trial. Just open the skull, do a craniotomy, you open the dura, and then inject cells and cover with Fibrin group, and that's it. So it is not very difficult to produce, to perform this kind of surgery. And the infarction volume has significantly reduced after this treatment. And the motor performance has been improved in rotator test and grasping power test. So based on this animal experiment, we further test adding growth factor together with bone marrow stem cell in treating a chronic focal cerebral ischemia. In this study, we used the GDNF to rescue as the growth factor to rescue the cerebral ischemia. The GDNF is a factor transforming growth factor beta family. It is a peptide protein which can rescue the dopaminergic neuron, motor neuron, or sensory sympathetic neuron. The GDNF gene expression in the early stage of focal brain ischemia has some effect, and topical application can reduce brain infarction and can reduce the brain edema, and it has been we have a previous study in the MCAO model using the GDNF, which showed a neuroprotective effect after the immediate application. That means the acute treatment. We have published in 2004. So this is the treatment protocol. First, again, do the MCA ligation and clipping, and then do the reperfusion 1 hour later, and follow up, and then treat it 1 week later. This time we add stem cell GDNF group to rescue the cerebral ischemia. So this is, as you can see, the effect seems to be better than the bone marrow stem cell with this group. And also, on the lateral view, see better regeneration of the cerebral vasculature on the surface of cortex comparing to animal received bone marrow stem cell group only. The infarction volume is also reduced, but not significant than other groups. They all show significant reduce of the infarction volume than the control, and there seems to be a little bit more further reduced than the infarction volume than the bone marrow stem cell together with Fibrin group. Neurodegenerative test, as you can see, both the GDNF treat bone marrow stem cell, GDNF together with bone marrow stem cell, also all 3 groups improve the locomotive behavior in the rotator test at 1 week, 2 weeks, 3 weeks, and 4 weeks. And grasping power, all 3 groups treated with GDNF bone marrow stem cell or GDNF together with bone marrow stem cell shows improvement of the grasping power 2, 3, 4 weeks after the transplantation. this is a windshield reaching test to test the dedicated function of the forelimb, the forepaw, which you can see put the foot part here, and animal should to reach to extend its forepaw to reach the foot palate and grasp it and to take it. And there are several scores to measure. And there is inaccurate reaching, or just touch it but cannot grasp it, or other area the animal cannot get full score. You can see only the animal receive bone marrow stem cell transplantation and GDNF treatment has a significant improvement in windshield reaching test. The GDNF treatment or bone marrow stem cell treatment has not shown any improvement significantly than the controls. So this is the most sensitive test to test the delicate movement of the forepaw after the transplantation. We work with the Professor Zhang in MSC to test MRI on the animal. This is the result. You can see after transplantation. And this is the animal without any transplantation. You can see the cerebral infarction and edema of the cerebral cortex. This group, you can see after the transplantation of the bone marrow stem cell and GDNF, you can see the reduce the ischemic area in the MRI. And this is functional MRI using the forepaw stimulation after transplantation. You can see the animal received the transplantation can get biologic active signals in the transplantation cortex. This is, we also test the cell with animal cells, human cells in the animal brain. You see positive both in the cell group and the cell together with GDNF group. And this is it. We stand with the human specific antibody. You can see the positive human cells here. And when we look at the immunohistochemistry, you can see we stand is human cell with BRBU. Trace this human cell's behavior. You can see the human cell in the penumbra migrate through the ischemic area into the cerebral cortex of rodent cerebral cortex. You can see the red dots indicating the human transplant stem cells into the rodent brain. And this is the MCAO with blue without stem cell. You can see there's no red dots, human cells inside the rodent cerebral cortex. After this experiment, we will form a team to conduct a human clinical trial on the subacute chronic cerebral ischemia. Now we are, because based on the clinical trial in human study, we have to form a comprehensive team, including neurosurgeon and neurology and neurorehabilitation doctors. In the Cerebral ischemia project, actually we have started to use RTMS and also other self-design method to rehab different kind of neurologic deficit after stroke. This together would put together with the cell therapy and growth factor therapy on the stroke patient. That will finally improve the functional outcome of these patients. The last I will show to send my acknowledgement to our colleagues. Hong Shijie, which is the PhD from the Tokyo University, which is the major contributor for the sensitive stem cell technology. Mei Zhen's PhD in my lab, and Su Fenjin is a PhD professor from Chengkong University in the differentiation model and sensitive model, and Huang Xiangsuo in Zhongshan University, Chang Shenguo in previously Xindian Tzu-Chi's the chairman. Now he moved to Zhenxin University, the vice superintendent. We have long-term collaboration on this stroke model, and also the Kaolang Shen, which is the director of the